Okay, so yesterday I think we got to neogli or the gli cells. Okay, and we learned that the gli cells are the protective cells of the nervous system. Okay, so that simply means that they play protective roles in the nervous system. Okay, so if you go to a system like the circulatory system, we have cells like the white blood cells playing protective roles. Now, when you come to the nervous system, okay, the nuclear cells plays protective roles, meaning they are more like caretakers. Okay, they are more like caretakers, or they protect the neurons in the nervous system. Okay, we also discussed that we have some nuclear cells in the central nervous system, and we have some nuclear cells in the peripheral nervous system. Okay, now when you come to the central nervous system, the glial cells that we have over there, we have the oligodendrocytes, we have the astrocytes, we have the ependymal cells, we have the microglia cells. And then in the peripheral nervous system, we have two neoglial cells. We have the squam cells, and then we have the satellite cells. Is that okay? Great. Now, we also mentioned that when we say oligodendrocytes, they form the myelin sheath for the neurons in the central nervous system. Okay, then we have the astrocytes, and we mentioned the astrocytes are the most abundant glial cells that we have in the nervous system, and they form part of the blood-brain barrier. Okay, again, we also spoke about the ependymal cells, and we mentioned that when it comes to cerebrospinal fluid circulating in the brain and spinal cord, the ependymal cells play a massive role. And those ones that are responsible for removing debris, pathogens, and damaged cells, they are the microglia cells. They are also within the central nervous system. Good. Then we also discussed the two neoglial cells in the peripheral nervous system, the squam cells and then the satellite cells. And we mentioned that the squam cells, just like the oligodendrocytes in the central nervous system, they also form the myelin sheath in the peripheral nervous system, meaning they form the myelin sheath for the neurons in the peripheral nervous system. Then we also mentioned the satellite cells. Satellite cells basically surround neuron cell bodies in the ganglia. Okay, so if you remember, we spoke about the ganglion. Satellite cell practically surrounds the ganglions. Okay, great. So these are the nuclear cells. Okay, so let's look at an image. So we have, practically this is how they look like. So this is the ependymal cells. Okay, this is the ependymal cells. This is how ependymal cells looks like. And these ones are the most abundant ones. This is how astrocytes look like. Okay, and these are all neoglial cells. So this is how the oligodendrocyte also looks like. Okay, so we have these four in the central nervous system: ependymal cells, the oligodendrocytes, the microglia, and then the astrocytes. Then in the peripheral nervous system, we have two. We have the satellite cells, and then we have the squam cells. Is that okay? Great. Yes. So take note. At least, basically, no, you should be able to state the or state some of the examples of neoglial cells in the central nervous system, or the four. You should be able to state the neoglial cells in the peripheral nervous system, or the two. Okay, and at least know about their functions, what they do. Okay, yeah. So practically, this is how they look like anatomically. Okay, that is fine. That is fine. So, and I think we went through. Did we go through the divisions of the nervous system?
Yes, sir.
Excellent. Great. That is fine. Yes. So, then that is good. Then it means it's possible we can finish in the next 1 hour 30 minutes. I think we should be able to finish the. Okay, that is fine. Now, let's move on to nerve impulses. Okay, now when we say a nerve impulse simply means, you see, remember I told you that for neurons or for nerves, when impulses are moving along the nerve, okay, or when electrical signals are moving along the nerve, we call it an impulse. So let me go back to the neuron diagram. So you see this neuron over here. When electrical signals are moving along the neuron, we call it a nerve impulse. Is that okay? That is what. So the movement of electrical signal from one point on the neuron to another point on the neuron. So this point A to another point B. That is what we call nerve impulse. But you see, in most textbooks, usually the axon of a neuron is very long. Okay, so if you look at the axon, you could see clearly that the axon is very long. So in most textbooks, when they are talking about nerve impulse, they will just highlight that a nerve impulse is the movement of electrical signal from one point of the axon to another point. But what you should know is that it's not only the axon that is forming the neuron. Okay, so nerve impulse simply means the movement of electrical signals from one point of the neuron to another point on the neuron. That is what we call a nerve impulse. Okay, but like I said, in most textbook or literatures, when we say nerve impulse, they would limit their definition to the axon. Okay, so the movement of electrical signals from one point on the axon to another point on the axon of the neuron. That is what we call nerve impulse. Okay, great. And for nervous system, nerve impulses travels at a very faster rate. So you see, a nerve impulse are electrical signals transmitted along the axon to communicate information. Is that okay? Yeah, so that is what I was trying to demonstrate over here. Okay, so the movement of electrical signals from one point to another point on the axon of a neuron. And the question is that why are electrical signals moving from one point of the neuron or the axon of a neuron to another point on the axon of a neuron? They are moving to communicate information. So anytime an electrical signal is moving along the axon of a neuron, it is carrying a certain information. So it's more like it's communicating a certain information somewhere. And that is why nerve impulses are very important. So as you are listening right now, as you are looking on your screen right now, impulses are being transmitted along neurons to certain parts of your brain for interpretations and processing to be made. Is that okay? And that's how interesting it is. Okay, yeah. So a nerve impulse simply means the movement of what? Electrical signals from one point on the axon of a neuron to another point on the axon to communicate an information. Is that, and that is very important for a lot of processes to go on in your body. Okay, great. So that is what we call nerve impulse. Okay, so let's assume this diagram you are seeing over here. Let's assume this is the axon. So this is point A, this is point B. So the movement of what? Electrical signals from point A on the axon to point B on the axon. That is what we call a nerve impulse. Is that okay? Great. Now, when we take a myelinated neuron and an unmyelinated neuron, which of them is going to have a very fast nerve impulse transmission?
The myelinated neuron.
Yes, excellent. And let me explain the reason why. Physiologically, let me explain to you the reason why that happens. Now, in the myelinated neurons, listen carefully. In a myelinated neuron, so this is an example of a myelinated neuron, the diagram you are seeing over here. In a myelinated neuron, the interesting thing is that it's like I'll explain it basically in a way to make it very simple for you. In a myelinated neuron, instead of the nerve impulse traveling along the axon completely, it travels only at the areas where the node of Rehnweis are. So it's sort of when it gets to where the myelins are, they jump. So let me go back to the diagram. Okay, so in a myelinated neuron, you see this is where this is how the myelinated neuron looks like. So you see the myelin sheets are here. So you see when the impulse ideally is supposed to travel along the entire neuron from here to here. Ideally, the impulse is supposed to travel along the entire neuron. But the interesting thing is with the myelinated neuron, it only travels within, the impulses are only transmitted within the node of Rein-Vine. So you see, so from here, the impulse will be coming from here. When it gets to the myelin sheath, it will jump to the node of Rein-Vine. When it gets to this node of Rein-Vine, it jumps to the next node of Rein-Vine. When it gets to this node of Rein-Vine, it will jump to the next. So at the end of the day, it makes the impulse transmission very fast. Now, in physiology, that process whereby for a myelinated neuron, the impulse transmission is only taking place within the node of Rein-Vine and having to jump over the myelin sheath. That is what we call saltatory conduction. In physiology, we call that saltatory conduction. Okay, and that is what I have in this diagram over here. I don't think I have this diagram in the slide I shared with you, but you can pay attention to it. So you see, with a myelinated neuron, okay, the impulse transmission only takes place at the node of Rein-Vine. So you see, so this is the node of Rein-Vine. When it gets over here, the impulse will jump over the myelin sheath. And that is what the jumping in physiology, that is what we call saltatory. Okay, and we call that saltatory conduction. So when we say saltatory conduction, it simply means that for a myelinated neuron or for an axon that is having a myelination or a myelin, is that okay? During impulse transmission, impulses are only transmitted within the node of Rein-Vine. When it gets to the point where the myelin sheets are, it jumps over. And that is what we call the saltatory conduction. Okay, yeah, so you see over here, we have, so the impulse transmission would only occur at this point. Okay, so as the impulse is going, when it gets over here, it will jump to the next point. Then the impulse transmission will happen again over here. When it gets to the myelin, it will jump. And that jumping process is what we call the saltatory conduction. Is that okay? Great. And that saltatory conduction only takes place in neurons that are having the myelin sheath. Okay, it's a very interesting process. Okay, great. Now let's look at impulse transmission goes through certain processes. But before we look at impulse transmission, let's first of all look at what we call potentials in the nervous system. Potentials in the nervous system. Okay, now when we say potentials in the nervous system, it's also a very interesting subject for you to understand. For you to understand nerve impulse transmission. Okay, now when we say a potential simply means the differences in electrical charge across a neuron. Okay, the differences in electrical charge across a neuron. Okay, and let me tell you, these differences in the electrical charge across a neuron, that is what helps it to generate and transmit electrical signals. Is that okay? So for electrical signals to be transmitted along or across a neuron membrane, okay, there should be some electrical signals or differences in the charges. Okay, so let's go to this diagram over here. So let's come back to this diagram. Good. Let's come back to this diagram. Remember, I told you that for impulse to be transmitted, okay, for impulse to be transmitted along this axon, the inside of the axon have to be positive. Okay, for impulse. So you see over here, look at this diagram over here. For impulse to be transmitted along this axon, the inside of the axon have to be positive and the outside have to be negative. But ideally, when the neuron is there, when it is at rest, no impulse is being transmitted. When it is at rest, the inside is negative. Okay, for impulse to be transmitted along the neuron, the inside have to become what? Positive. Please understand this principle before we move on. Okay, I think I have a new diagram in here. Let me see if it is there. Okay, great. So you see, this is a beautiful diagram. Okay, so this is the axon. This is the axon. Where I'm pointing now is the axon of the neuron. This is the axon of the neuron. Now, when a neuron is at rest, the inside is positive. So you see here, the inside is negative. So you see here, the inside of the neuron is negative. Is that okay? This shows that this neuron is resting. No impulse is being transmitted. Is that okay? Now, when the inside is negative, that should also tell you the outside is what? The outside is what? Is positive. Okay. Now the difference is, so you see the difference in the charges that we are talking about is here. This and this. Okay, is the vertical difference. Okay, so if the axon is resting, the inside is what? Is negative, and the outside is positive. So you see the difference I'm explaining now is the vertical difference. The vertical difference. Now, that vertical difference that is explaining the charges is what we call the membrane potential. Okay, so let me explain again. So the axon is this. This is the axon of the neuron. From here to here is the axon. So you see the impulse transmission is coming from where? The dendrite. The electrical signal is coming from the dendrite. Now, when the electrical signal gets over here, just follow the arrow, what I'm doing with the cursor. When the signal gets over here, okay, before the impulse will be able to move from here to here, okay, the positive charges here have to come inside for the negative to also go outside. Okay, once that happens, then the impulse will now move to the next level. When it moves to the next level, when it moves to this level, the positive that is outside at this level, the positive outside here, have to now move inside for the negative at that level to also what? Come outside. Okay, so you see that difference in the charges that are created for the impulse to move, that is what we call membrane potential. Okay, now the movement of the impulse itself in a horizontal way along the axon, that is what we refer to as the action potential. Is that okay? So like this, when the impulse is moving in this direction, when the impulse is moving in this direction, that is what we call the action potential. But you see, before the impulse will move in this direction, there should be a membrane potential in this vertical direction. Okay, so because you see, at the membrane over here, okay, the outside, this area is positive and this area is negative. Now, before the impulse will move from here to here, the negative over here have to come outside for the positive to come in. Then the signal will now come to this level. For the signal to continue moving at this same level, the negative at this level, the positive at this level have to come inside for the negative at this level to also go outside. Then the impulse will continue. Now, when it gets to this point, the negative over here, the positive over here have to come inside. Then the negative will now go outside. Then the impulse will continue again. When it gets to this point, the positive here will now have to come inside. Then the negative here will now have to go outside. Then the impulse will continue. When it gets to this point, the positive here will then again come inside. Then the negative here would go outside. Then the impulse will continue like that. So practically, you see that the action potential depends on the membrane potential. Okay, let me start all over again. So for the impulse transmission is going this direction. Okay, from here to here. So the impulse transmission is going this direction. And that is what we call the action potential. So the action potential is moving this direction. Okay, and it's very interesting. Okay, but so let's assume the potential, the action, the impulse has gotten to this point. The impulse has gotten to this point. Now remember, if no impulse is being transmitted along the axon, the inside is negative and the outside is positive. Now, when the impulse gets over here, the moment the impulse gets over here, for the impulse to move from this point to this point, okay, at this level, the outside of the axon here is positive. The inside here is negative. There should be an exchange. The positive here have to come inside and the negative here will move outside. Then the impulse will move to this level. Once the impulse gets over here, the same principle. Once the impulse gets over here, the same principle. The positive here have to come inside. Then the negative will also come outside. Then the impulse will continue. Then the impulse will now continue. Once the impulse gets over here, the positive here will come inside again. The negative will come outside. Then the impulse will continue. Once the impulse gets over here, the same principle. The positive here will come inside. The negative will go outside. Then the impulse will continue like that. Okay, so practically you would agree with me that the action potential or the impulse transmission depends on the membrane potential. Okay, so when we say a membrane potential is like the electrical difference. Is that okay? So you see, the membrane potential is the electrical difference between the inside of the axon and the outside of the axon. Some books will say the electrical difference between the inside of the neuron and the outside of the neuron. That is what we call the membrane potential. Okay, but the action potential is the impulse transmission along the axon. That is the action potential, or the horizontal movement of the impulse along the axon. That is what we call the action potential. Is that okay? Great. Have you gotten it, or you want me to go over it again? Have you?
Please go over it
Take
again.
it again.
Okay, that is fine. So I'm going over it again. So this is what we call, this is the axon. This is a neuron. The diagram you are seeing over here is what? It's a neuron. And this portion is the cell body of the neuron. We have the dendrites over here. And typically this is the axon. We have the axon terminals over here. Okay, now the impulse, electrical signal will start coming from the dendrites. So the impulse will get to this point. Now, for the impulse to be transmitted, the transmission or the longitudinal or horizontal transmission of the impulse along the axon, that is what we call the action potential. Okay, so the transmission from here. So if the impulse have to move from here to here, that will be possible through the action potential. That is what we call the action potential. Okay, so from the impulse being transmitted, or the electrical signal being transmitted from point A here on the axon to point B here on the axon, that is what we call the action potential. Okay, that's more like the horizontal transmission. Okay, now before the impulse will be able to move to this direction, it goes through certain processes. So it will start from here. Now remember that when the axon is at rest or a neuron is at rest, the inside is negative, the outside is positive. When a neuron is at rest, the inside is negative, the outside is positive. Okay, now before the impulse will move from here to here, okay, the outside here, which is positive, have to move inside. Then the negative charge inside have to move outside. Once that occurs, then the impulse will move to the next level. Now, when the impulse gets to this level, it has to continue moving. Now, over here to the outside is positive, the inside is negative. So then exchange will take place again, so that the positive outside will come inside, then the negative inside goes outside again. Once that happens, impulse will continue moving. When the impulse gets to this point, the same principle. The positive outside will move inside, the negative move outside, then the impulse continues. Now, once it gets to this point, the positive outside here also move inside, the negative also move outside, then the impulse continues. Okay, so you agree with me that following what I'm doing right now, you agree with me that for the impulse to move along the axon, there should be exchange in the charges across the membrane. Now, the differences between the charges in the inside of the neuron and the outside of the neuron, that is what we call the membrane potential. So when we say membrane potential simply means the differences between the electrical charges in the inside of the neuron and the outside of the neuron. Okay, and when we say an action potential, it's simply the transmission of the electrical charge along the neuron. That's the action potential. the transmission. Okay, so let me do it again. When the impulse gets over here, before it moves to the next level, the positive charge at the outside have to come inside for the negative charge inside to go outside. Then the impulse will move. When the impulse moves to the next point, the positive charge there have to move inside. Then the negative charge move outside. Then the impulse continues again. Okay, so that difference between the inside, the electrical difference between the inside and the outside, that is what we call the membrane potential. Have you gotten it now? Have you gotten it now? Who can explain? Yes, who can explain? Yes, who can explain?
So you said that for an impulse to move to another level,
another point on the neuron.
Another point of the neuron. The positive outside has to come inside.
Good.
And then
Excellent.
the negative inside has to go outside.
Excellent.
And
so
then
once
continue
on. I'm listening. Continue on. Continue on.
And you said that the movement of impulse along the axon is the action potential.
Excellent. Good.
And then the electrical difference between the inside of the neuron and outside of the neuron is the membrane potential movement.
That is excellent. This shows that you've understood the principles. Is that okay? So if I ask you, okay, so which of them depend on the other?
The action potential depends on the membrane potential.
That is excellent. The action potential depends on the membrane potential. This shows clearly that you've gotten the principle. So you see, the thing is like this. So you see, when the impulse gets here, the signal is coming from the dendrites. When it gets here, before it will be transmitted along the axon. So you see, the impulse getting to be transmitted along the axon is what we call the impulse getting to be transmitted along the axon. That is what we call the action potential. Okay, so the impulse having to be transmitted from here A to here B, that is what we call the action potential. But before the impulse will get to this point, a lot of things will have to change at the inside and outside before the impulse move. So it's like this. Okay, so this is how it goes. This is how it goes. So when the impulse gets here, at this level, the neuron is at rest. So when the impulse gets here, before the impulse move one step, the positive outside have to move inside, then the negative inside move outside, then the impulse move one step. Okay, now once the impulse move one step, it has to continue. So where it has gotten to, the positive there have to move inside for the negative there to move outside, then the impulse move another step again. Is that okay? When it gets over here, the positive here have to move inside again, then the negative move outside, then the impulse move another step. And this thing we are explaining like this is happening at a faster rate. It's happening at a very fast rate. Okay, now another additional thing that I'll add to it is that, listen carefully. Another additional thing I'm going to add to it is that, don't make it confuse you, is that now the outside, you see, we mentioned that the outside is positive. When a neuron is at rest, the outside is what? Positive, and the inside is negative. Now, what makes the outside positive is that we have sodium at the outside. We have a lot of sodium outside the neuron, and we have a lot of potassium inside. Okay, so what that means is that for the impulse to move, potassium have to go outside for sodium to come inside. It's the same thing. We've not changed any principle. Listen carefully. We said that when the neuron is at rest, the inside is what? Negative. The outside is positive. Now, if impulse have to move or action potential have to occur, okay, the outside that is positive have to come inside for the inside to become positive. Then the negative that is inside have to go outside so that the outside become negative. Is that okay? So that means that before impulse transmission would occur, the inside of the neuron have to be what? The inside of the neuron have to be positive and the outside have to be negative. That is what we mean. Now, and I'm adding that what makes the inside negative is that we have potassium inside. And what makes the outside positive is that we have sodium outside. So this is how it works. So you see, when the impulse gets here, the sodium over here will move inside, the potassium will move out. Then the impulse will move. It's the same thing, just that now I'm using the electrolytes. Now, when the impulse moves this step, the sodium at this point will now move inside. The potassium will move outside. Then the inside become positive. Then the impulse will move. Is that okay? Then it moves another step. When it gets to this step, the sodium here will move inside, potassium move outside, then the impulse move. When it gets to this level, the sodium over here will move outside, okay? Will move inside, the potassium will move outside, then the impulse will continue. When it gets over here, sodium outside here will move inside, potassium will move outside, then the impulse will continue. When it gets over here, sodium at this level will move inside, potassium will move outside, impulse continue. When it gets over here, the sodium here will move inside, potassium goes outside, impulse continues. Is that okay? So you see, the electrical charges also depends on the potassium and sodium, because it's the potassium inside that makes the neuron negative, and it's the sodium outside that makes it positive. So for the impulse to move, there should be exchange of the potassium and sodium. The potassium have to move outside, the sodium have to move inside. Please get these principles. Is that okay? Or simply put, for the impulse to move or action potential to occur, the sodium, the positive charge outside have to move inside, and the negative charge inside have to move outside. Is that okay? And these are very simple principles that you have to know. Okay, so now let me ask you this question. What is the difference between action potential and membrane potential? What's the difference between action potential and membrane potential?
So please, can I try?
Yes, please. Please
Yeah.
try.
The action potential is the movement of electrical signals from one point to another point in the axon. And the
That
memory
is excellent. Yes, I like your definition a lot.
and the
Yes.
memory potential is the electrical difference between the inside of the neuron and the outside of the neuron.
Okay. So which one depends on the other?
The action potential depends on the memory potential.
Can you explain that?
Yes, sir.
Okay, explain.
Yes, because before the signal can transport from the inside the axons, it depends on the memory potential, because the electrical signals will be changing.
Okay, excellent. Good.
Good.
The electrical signals have to be changing from
Positive to negative.
yes,
and
from positive to negative, negative to positive. As that happens, then the impulse is moving. Excellent. That is brilliant. Is that okay? That is brilliant. Okay, so the sodium have to be coming inside to make the neuron positive, and the potassium have to be going outside to make the outside negative. Once that happens, then the impulse will move. Then for the impulse to continue moving, the outside that is positive have to come inside to become positive. Then the inside that is negative have to go outside. Then the impulse continue moving. That is how interesting it is. Okay, so you can imagine, and all these processes are going on in our bodies as you are moving, as you are viewing objects, as you are hearing. All these processes are going on in your body. Now, when a neuron is at rest, is that okay? That is what we call resting membrane potential. When a neuron is at rest, in physiology or in anatomy and physiology, that is what we call resting membrane potential, meaning there is no action potential. Because you see, action potential only occurs when impulse is moving. Okay, but when a neuron is at rest, we say that the neuron is at a resting membrane potential. Okay, and if a neuron is at rest, the resting membrane potential is negative 70 millivolts. Is that okay? When a neuron is at rest. Okay, there is a graph over here. Good. So you see, here is it. So here, this neuron is at rest. No impulse transmission. So you see, there is no impulse transmission. So the neuron, when the neuron is at rest, the voltage is negative 70. Is that okay? Negative 70. Okay, now, when the impulse, the voltage rises up to negative 55, then we say that the neuron have gotten to a threshold, and we call that threshold potential. Okay, so when we say threshold potential, meaning the neuron have been stimulated. You remember neurons can be excited. If you remember, I taught you that one of the properties of a neuron is that it can be excited, excitability. Okay, so when a neuron is excited, when it is at rest, when it gets to negative 55 volts, that is millivolts, okay, then it has gotten to a threshold. What that means is that when it hits the threshold, it's going to depolarize, meaning an action potential is going to occur. When we say the neuron is going to depolarize, meaning an action potential or movement of an impulse is going to occur. Is that okay? So the moment a neuron is at negative 70 millivolts, it's resting. When every neuron is at negative 70, it's in a resting state. Now, in a resting state, what is the membrane potential? Who can explain? In a resting state of a neuron, what is the membrane potential?
Inside negative, a positive, a negative.
That is excellent. Yes, the
Yes.
inside is negative, the outside is
Negative and
positive.
outside is positive.
Yes, that is excellent. But in terms of electrolytes, how will you explain it? In terms of using
So
the sodium and potassium?
the potassium will move inside and then sodium will come outside.
That is excellent. Potassium will be inside and sodium will be outside. It shows that you are concentrating. So now let's move on to the next step. Now, when the neuron is being stimulated, now it will start moving towards the threshold. It's like, those days when you are going to do racing, you see, they will nail down, then they will say on the max. It means you are going to fire. Once you hear you fire. The threshold is like the neuron has been stimulated at rest to the point that now it's coming to shoot. It's like we are telling the neuron on the max. So it has charged. It's now coming to that. Then we say it has gotten to the threshold. Is that okay? That is when we say that it has gotten to a threshold. But the threshold doesn't mean there is an action potential. But when it gets to the threshold, any other stimulation will make it depolarize. So it's like we've told you on the max, you are kneeling down. Once you hear pa, you move. Any other sound that you hear is going to cause you to move. That's the same principle. It's something like that. Okay. So when a neuron is at rest, it is negative 70. When it reaches the threshold at negative 55 millivolts. So the moment it reaches the threshold, the next thing is that it's going to depolarize. Now, if it is going to depolarize, what it means is that impulse is going to move. So when we say a neuron has depolarized, meaning impulse is moving, meaning action potential has occurred. Let me go over that again. When we say that a neuron has depolarized, meaning an action potential have occurred, meaning an impulse have moved along the axon. Now, my question is that from this explanation, if depolarization means an action potential has occurred, what would be the membrane potential at depolarization? What would be the membrane potential? What would be the difference between the charges on the neuron when it depolarizes? When the neuron depolarizes, when we say that when it depolarizes, impulse is moving along it. So what will be the differences between the charges? What will happen to the inside and what will happen to the outside?
So the inside will be
positive and the outside will be negative.
That is simple. And that is what has been shown over here. So you see, sodium ions move in. That is what has been shown over here. It means that sodium, and remember I told you, if sodium ions move in, then the inside becomes positive. So sodium ions will move in. So during depolarization, sodium ions will move in. What will happen to potassium? Potassium will do what?
Will move outside.
Potassium will move outside. So during depolarization, the sodium ions will now move in. Potassium ions move out. Is that okay? Okay. When the neuron is at rest, what happens to the sodium and potassium? Let's start over. When the neuron is at rest, what happens to the potassium and then sodium?
So the
Sodium will be outside and potassium will be inside.
Will be
Excellent.
negative and the potassium
Excellent.
will be positive.
Excellent. Excellent. Now, the moment it gets to the threshold, then it depolarizes, meaning it fires. When it depolarizes, it means that now the outside has now become negative and the inside is what? Is positive. Okay, so it will fire till it gets to the peak. The peak is somewhere around positive 40. That's the peak. Once it gets to the peak, meaning it has fired, it now have to come back home. It has to come back to rest again and fire another one. Okay, and it's coming back. So you remember I told you that, you see, don't assume that the sodium, the positive comes inside once now, then the impulse will go and reach the axon. No, it's occurring gradually along the axon. So it starts from here. Okay, so the positive come inside, the negative go outside. It move one. Is that it will go back to rest again. Then the process will start all over again. That is what we are explaining with the graph. Okay, so here it was at rest. Sodium was outside, potassium was inside. It was resting, no impulse transmission, no action potential. Now it reaches the threshold. Now, when it gets to the threshold, the next thing is it's coming to fire. That is what we call depolarization. Now, for it to depolarize, sodium will now move inside, potassium will move outside. Then it will fire till it reaches the peak. When it reaches the peak, means here impulse has been transmitted. Once impulse has been transmitted, it has to come back to rest again. Now, the process of the impulse or the signal coming back to rest, that is what we call the repolarization. But there is something interesting over here that I want you to learn for once. There is something interesting that happens over here. Now, it is now coming to rest. Okay, look at this. It is now coming to rest. Ideally, what are you expecting to happen to sodium and potassium? What are you expecting to happen to the sodium and potassium? Yes, who can give us the explanation? Let's have one person. Ideally, we mentioned that before it fire, sodium have to come inside, potassium have to go outside. Now it has finished firing, meaning an action potential has occurred. It's going back to sleep again. What should happen to potassium and what should happen to sodium?
So it will
Potassium will come inside and sodium will go outside.
Potassium will do what?
go
Will come
inside.
inside.
And sodium will go outside.
That is excellent. I like the way you are thinking like a natural person. But in physiology, at this point, during the repolarization, the potassium will remain outside, but the sodium will move outside. Listen to it carefully. In physiology, during the repolarization phase, what happens is that the potassium will remain outside, but then the sodium will come outside. Now, in this situation, the inside will still be negative. Keep it in your mind. During the repolarization phase, the potassium will remain outside, but the sodium will move outside. In this situation, the inside will still be negative. Keep it in your mind. That is what happens in the repolarization phase. And it confuses a lot of people because, you see, per natural thinking, you're expecting that once the cell is going to rest, potassium should come inside and then sodium goes outside. Okay, but what happens during the repolarization is that the sodium will move outside, but the potassium remains outside. Okay, potassium remains outside. That is what happens. Is that okay? So keep that in your mind. Okay, good. So, yeah, so that's what happened. So that's what
Sir,
happens. Yes.
please, I have a question.
Yes, ask your question, please. Ask your
Sir,
question.
you said in physiology, the
Yes.
sodium has to move outside and the potassium
Yes.
remains outside, but the inside
Yes.
still becomes negative.
Yes.
Please, sir, how is that?
What makes the inside negative is the potassium. Okay. And what makes the inside positive is the sodium. Now look at the situation. If the sodium is not inside, the inside will still be negative even without the potassium. Okay, so because listen, what makes the inside positive? Yes, what makes the inside positive?
Because of the sodium.
No, what makes the inside of the neuron positive?
When
What makes the inside positive?
The presence of the sodium.
Come outside.
When will the neuron, the inside, become positive?
Transfusion.
Oh no, the answers you are giving are completely off. What will make the inside become positive?
When urine is at work.
No. What will make the inside of the neuron to become positive?
When the impulse is passing through.
Transfusion.
It's not true. When
When the
sodium moves into the neuron, why are you giving off
So
answers?
that's what we said.
When the sodium moves into the neuron, that is when the inside become positive. So listen carefully. If sodium is moving outside, will the inside be positive?
no.
The
Most
It
inside
will
will
still
not be
be
positive.
negative.
It will still be negative.
Yes.
So it's not necessarily about the potassium. Even though when the inside is negative, we are expecting potassium to be there. But what makes the inside positive is the sodium, not the potassium. Okay, so the moment we are able to get the sodium to move outside, even if potassium don't come inside, still the inside will be negative. If you don't understand that, it will be difficult for you to understand the repolarization. So with the repolarization, what happens is that even though naturally you'll be expecting that, listen, if in the repolarization, you'll be expecting that because the neuron is going back to rest, the potassium have to come inside and the sodium have to go inside. It doesn't happen that way. In the repolarization, the sodium go out, potassium remains out. Keep it in your mind.
So
In the
both.
repolarization, yes.
Okay.
Someone wanted to echo something. So
No,
both,
I was asking. So meaning both the sodium and potassium will be outside the
both
neuron.
of them remain outside in repolarization. Keep that
All
in
right.
your mind. Okay, both of them remains outside. Now, and that is what I was trying to explain, that what makes the inside positive is the sodium. So once we get sodium to go outside, we've achieved the negative. It's not about potassium coming in. What makes the inside negative is when the sodium is outside. Then automatically the inside will be negative. So during the repolarization phase, what happens is that, okay, sodium will go outside or sodium will exit. Is that okay? Sodium go outside, but potassium remains outside also. Is that okay? That is what happens with the repolarization. Okay, then the cell will gradually be moving towards the negative, gradually. Because with the repolarization, we want the cell to return back to rest gradually. Is that okay? That is what we are expecting with the repolarization. Now, so the cell gradually comes back to rest at this level. But we can have a situation that we call the hyperpolarization. Okay, the hyperpolarization is a situation where more like the potassium channels remain open, then more of potassium can go inside, and the cell or the neuron will become highly negative. We call that hyperpolarization. Is that okay? Yes, and that can happen sometimes, especially when we want to start a new action potential. Hyperpolarization can occur. Now, the two areas I want you to focus on is that at least know what happens when the neuron is at rest. So I'm going to gradually take you through that. When the neuron is at rest, what happens to sodium and potassium? Let's take it gradually. When the neuron is at rest, what happens to potassium and sodium?
Sodium will be outside and
Potassium will be moved inside.
That is excellent. Now let's move on to depolarization. Now, for the neuron to depolarize, what should happen to sodium and potassium for the neuron to depolarize?
Sodium has to move in
Sodium
for
has
the
to go
potassium to
outside
move
and
out.
potassium has to also remain outside.
Hey, madam, you missed it. Oh, madam, you missed it. You missed it. That is not depolarization.
The
Go
sodium goes
inside,
inside
potassium has
and
to
the
move
potassium
outside.
comes outside.
Now giving the correct answer. Yes. So sodium have to move inside, potassium have to move outside. Is that okay? Okay, now let's go to the one that confuses a lot of people. With the repolarization, what happens?
The potassium remains outside and the sodium too comes outside.
That is excellent. That is brilliant. Once you are able to
Outside,
show
and
that,
sodium will
eh,
rather move outside.
good, excellent. That shows that you've understood it. So during the repolarization, don't go and say that during repolarization, potassium will move inside. No. During repolarization, the potassium remains outside, but the sodium comes outside. Is that okay? Yeah, then gradually the cell comes back to what? To rest again. Then a new action potential will begin. Is that okay? Yes. So this is what
Yes.
we call action potential in nervous system. Is that okay? This is what we call action potential. Now, this action potential is more of physiology things. Now, if I'll ask you something over here, the only thing I can ask you questions on is the membrane potential. So I can ask you a question like, for depolarization to occur, what will happen to sodium and potassium? Then I'll bring sodium in, potassium out, those kind of things. Okay, so you have to pay attention. Okay,
Sir, please
the
can you
same thing.
go over the threshold?
Come again.
Please can you go over the threshold?
With threshold, okay, so threshold simply means that the neuron have been excited to a point that is the depolarization is coming to occur. When we say threshold simply means it has reached a point where an action potential is coming to occur. And remember, we learned the key properties of a neuron, that a neuron can be excited. Okay, so if a neuron has not been excited and it is there, now it means that the neuron is at rest. Now, at resting stage, the voltage is negative 70 millivolts. Okay, now, if a neuron is being excited, once the neuron is being excited, it will get to the threshold potential. When we say threshold potential, what it means is that it has gotten to a certain point that an action potential is going to begin. That is what we call threshold potential. So right after the threshold potential, an action potential begins. The neuron, the impulse will shoot, or an action potential will now shoot. And that is where depolarization comes in. So it starts from where the neuron is at rest. Then a stimulus will come in. Then the stimulus will push it to the threshold potential. From the threshold potential straight, it depolarizes. Now, for it to depolarize, sodium have to move inside the neuron. Potassium have to move outside. That will happen until it will get to the peak. Once it gets to the peak action potential, the peak action potential too is about positive 40 millivolts. Once it gets over here, then repolarization has to occur. But there is something unique about the repolarization that you have to get. Now, the principle is that for repolarization to occur, potassium ions will remain outside, but sodium ions come outside. That is what we call the repolarization. Is that okay? Great. Then the cell gradually comes back to rest. Is that okay? Yes. And this process is what we call action potential. So if someone asks you that, okay, explain action potential, you are going to say this, okay, an action potential, or explain potentials in nervous system. Then you tell the person that, okay, potentials, we have two types of potentials. We have membrane potential and we have action potential. A membrane potential simply means the differences in the charges across the membrane of the neuron. That's the membrane potential. And the action potential simply means the movement of the impulse along the axon of a neuron. Now, the action potential depends on the membrane potential. That means that for an impulse to move one step, the outside that is positive have to move inside for the inside to become what? Positive. Then the outside will become negative. Then the impulse transmission will occur. Now, for another impulse transmission to occur again, okay, the outside at that level have to move inside so that the inside become positive. And that is what happens for action potentials to be generated. Okay, then again, you add that action potential goes through some stages. We have the resting state where no impulse transmission occurs. And at that state, sodium is at the outside and potassium is within the cell. Now, when a cell is resting and is being stimulated, for a long time, it gets to a point where it will reach the threshold potential. Now, the normal threshold potential is negative 55 millivolts. Once the neuron reaches the threshold potential, okay, automatically depolarization have to now occur. Okay, now for depolarization to occur, the sodium that is outside have to move inside the neuron for the negative or the potassium inside to move outside. Then the depolarization occurs until the neuron reaches the peak. Once it reaches the peak at negative 40 millivolts, is that okay? Then the cell have to come back to rest through a process called the repolarization. Then you add that, but there's something unique about the repolarization. Even though as the cell is going back to rest, we are expecting potassium to move inside, but during the repolarization phase, potassium remains outside, but sodium joins it outside. Sodium moves outside to join the potassium, and that is what gives us the repolarization. Then through the repolarization, gradually, the cell go back to rest again. Is that okay? That is simply what we call the action potential in nervous system. Okay, great. Can we have someone to summarize everything? The membrane potential, action potential, and the graph. Can we have one person to summarize? Yes, can we have one person, one of you, to summarize the action potential before we move on to the next stage?
So, can I try?
Yes, please. Please try. Try.
Okay. So please, you have two types of potentials. You have the membrane potential and then action potential. And then the
Excellent.
action potential is the movement of impulse along the atom.
All
And the
right.
membrane potential is the difference between the electric charges that moves across a neuron membrane. So with the graph, when the when the impulse is at rest, it's negative 70 millivolts. And when it
Okay.
gets to the threshold, it means that the neuron is excited. And that
Okay.
one is negative 55 volts. And when it
Okay.
reaches that state, it means it is ready to be depolarized. And
Okay.
what happens with the depolarization is that sodium moves in for potassium to move out.
Okay.
So
That's
when it reaches this, it moves at a peak. And when it gets to that peak, that means it's ready to repolarize.
okay. That's fine.
And what happens in the repolarization is that potassium remains outside for
Okay.
sodium to move outside.
Okay.
Then gradually it comes back to the resting stage.
Okay. That is fine. You didn't talk about hyperpolarization.
The hyperpolarization is when the potassium channel opens for more potassium. Sorry,
That
more
is great.
potassium to
So
move
that means
inside.
that during the hyperpolarization phase, that is a phase where the neuron cell can become more negative, highly negative. Okay. And you see, when hyperpolarization occurs, it means the action potential that is coming is serious. Because you see, the more it goes down to rest, the more the action potential that will be generated will be very higher. That's how neurons work. Okay, and there are times that a neuron can go to rest and it will even hyperpolarize. Is that okay? Hyperpolarization means that a lot of potassium would enter into the cell. Okay, a lot of potassium will enter into the cell for the inside of the neuron to become more negative. Is that okay? Yes. So I think you've done a very good job. It shows that you are concentrating. It shows that you are concentrating. So I mentioned that I can ask you few questions over here. The questions I can ask you over here is that for a neuron to depolarize, what will happen to sodium and potassium? Then I'll bring A. Sodium will move what? In, potassium will move out. Then B. Potassium will move here, sodium will do that. Those kind of questions. Okay, so here, pay attention to the resting. You can write it down. Pay attention to you. You can get questions on when the neuron is at rest. What happens to the sodium and potassium? Or what happens to the charges inside and the charges outside? You should expect questions on that. Now, during the depolarization phase, what happens to the sodium? What happens to potassium? During the repolarization phase, what happens to potassium? What happens to sodium? Is that okay? Kindly note it down. Okay, great. So those are the things that, fine. If you want to add on to the hyperpolarization, it's a plus for you. So during the hyperpolarization phase, what happens to what? What happens to the potassium? Now, the hyperpolarization is just about potassium. So with the hyperpolarization, we are going to have a lot of potassium move into the cell. Simple as that. Is that okay? All right, that is fine. So that is what we call action potential and then membrane potential. So I'm so happy that you've gotten the concept of the potentials in nervous system. Okay, great. Now let's do the reflex action in nervous system. Okay, let's do reflex action in nervous system. Okay, good. Now, when we say a reflex action, okay, a reflex action is an automatic, a rapid, and an involuntary response to a stimulus. Okay, and it's a system that is there, or a process that is there to protect your body from harm or your body from an injury. Is that okay? Yes, and we have something we call a reflex arc. A reflex arc is like a pathway that mediates the reflex action. Okay, so that neural integration or processing functions will not have to go and take place inside the brain. It will just occur at the level of the spinal cord. That is what we call a reflex action. Okay, now let's look at how a typical reflex action occurs. Pay attention here. Let's look at how a typical reflex action occurs. So for a reflex action to occur, look, we have, so this is the skin. Let's say this is the skin of an individual. This is the skin. So you see, within the skin, we have a receptor over here. And the receptor is a neuron. Oh, please pay attention. The receptor is a neuron. You see, the receptor is a neuron. So you see, we have a receptor over here. What type of neuron is this? What type of neuron is this? This neuron is taking information from the skin to the spinal cord. What type of neuron is this?
Motor neuron.
Hey, who said motor neuron? A neuron
Sir,
that
it's
is
a
taking
unipodal.
hey, a neuron that is taking an information from the skin to the spinal cord. That is a sensory neuron.
It's a sensory neuron.
Yes, that's a sensory neuron. Yes, that is a
Yes,
sensory
sensory
neuron.
neuron. Sorry.
Yes, so you see, this is the skin. This is a receptor in the skin. Okay, so this is taking information from the skin to the spinal cord. It's a sensory neuron. Is that okay? It's a sensory neuron. Okay, good. So you see, this is how a reflex act. Okay, so the neuron here, the sensory neuron, will pick the information. Now, which part of the neuron do you think is picking the information? Which part is this? Which part of the neuron typically will you call the dendrite? Is that okay? The dendrite. The dendrite. So it will pick the information. So you see, the information will be moving like an impulse. And all the action potential we discussed will be occurring now. Interesting. So you see, the inside. So before the impulse will be moving to the spinal cord, the sodium have to move inside, potassium outside, and the impulse will be moving. That will be occurring till it gets to this point. Is that okay? So now when it gets over here, the impulse will be released to the interneuron or the relay neuron. Now, what are the functions of a relay neuron? What are the functions of a relay neuron? Or what are the functions of interneurons?
It processes information.
Instantly, it processes information.
They make decisions.
Excellent. Yes, this shows that you people
Responsible.
are learning. Yes, it shows that you are learning. Yes, so it is responsible for processing informations, integration. So all the integration. So for example, someone have pricked your skin with a pin. Your sensory neurons will pick the signal and send it to the spinal cord. When it gets to the spinal cord, it relays it to what? The relay neuron. Or it transmits the signal to the relay neuron. The relay neuron will now integrate, meaning it will process and interpret. Do you get it? It will process the information, then say that, okay, let's send an information back to that part of your body to move it away from the prick. Is that okay? So you see, it will send the information to that part of the body. Which neuron is this? Which neuron do you think will be sending the information to the part of the body?
The motor neuron.
This time, this is the motor neuron. Is that okay? The motor neuron. This time, this is the motor neuron. Is that okay? Yes, so the motor neuron will now take the information to the part of the body, and it will take it to that part of the body that will take the action. Is what we call the effector. Okay, so in a typical reflex action, we have some structures that we are interested in anatomy. In a typical reflex action, we have the receptor, okay, which is more like the sensory neuron. Then the sensory neuron will relay the information towards a relay neuron. Then we have the motor neuron. Okay, then the motor neuron relays the information to the effector, which can be a muscle. Let me start all over again. And when I finish, I'll let 2 of you or 3 of you to explain. In a typical reflex action, okay, so let's assume it can be a fire. Let's say you step on a fire or a hot object. Your sensory neurons, the receptors attached to your sensory neurons will pick the signals. Then the signal will be transmitted along the sensory neuron, and then it will release the signal or the impulse to the relay neuron. The relay neuron will process the information and give a response, and release the response to the motor neuron. Then the motor neuron also release the information to the effector for the effector to take an action. Let me go over it again for the last time. Let's say your hand touches a hot object. In a situation like that, the sensory receptors in your skin attached to your sensory neurons will pick the signal. The signal will be transmitted along the sensory neuron, and the information will be sent to the relay neuron. The relay neuron will be the one to process the information within the spinal cord. Okay, so you see here, the information is not going to the brain. The relay neuron processes it within the spinal cord. Then it relays the information to where? To the motor neuron. Then the motor neuron sends the information to that part of the body there, and we call that part the effector, which can be a muscle for an action to be taken. Okay, now who can explain what I've just explained? In a typical reflex action, what happens?
Please, I want to try.
Yes, try.
So please, in the action where a knife cuts someone,
Okay,
the sensory receptors in the skin will send the message, will take the message to the sensory neuron,
okay,
for the sensory neuron to send the message to the relay neuron to interpret and process the information in the
okay,
spinal cord.
that's fine.
Then the relay neuron will relay the message to the motor neuron to take it
Okay,
to the part of the body.
okay,
Then the motor neuron will send it to the effector for him to act or for an action to be taken.
okay, that is fine. Can we have another person to also try? Yes, can we have another person? Yes, let's have another person to also try. You've done very well, Bless, Lady Bless. You've done well. Can we have another person? Hello.
Hello, sir.
Yes, can we have another person to also try?
Yes.
Okay, please, I want to try. Okay, so in the situation where there is fire and maybe a part of the body or maybe the hand touches it,
Okay,
so
that is fine.
it will send information to the receptors of
Okay,
the
that is great.
skin, and
That is
then
great.
it will send the information through the sensory neuron to
Mm-hmm.
the spinal cord where we will find the relay neuron, and then
Okay,
it will process the information, and then it will pass through the motor neurons,
okay,
and then it will go. It will go to the effector where there can be muscles in the body.
that is excellent. That is great. That is great. So you should be able to describe this process, and please learn how to draw the reflex arc. Okay, learn how to draw it very well. It's a typical area we can examine you on. Okay, so the reflex arc, learn how to draw a typical diagram of the reflex arc. Is that okay? Great. And a question we can ask you here in anatomy is that we can ask you to list all the structures that are involved in a typical reflex action. Okay, so in a typical reflex action process, what are the structures that are involved? So then you say, okay, we have the stimulus, we have the sensory neuron, we have the relay neuron, we have the motor neuron, okay, and then we have the effector. Please say it after me. Say it after me. The stimulus. Say the stimulus.
The
The
stimulus.
similar.
Neuron.
So the network.
Sensory neuron.
Okay. Then we have the relay neuron or interneuron.
The
Neuron or interneuron.
Say motor neuron.
Motor neuron. Effector.
Stimulus.
Stimulus. Sensory neuron. Relay neuron or interneuron.
Neuron.
Motor neuron.
Effector. Say effector.
effector.
If I ask you that, okay, in a typical reflex action, state the 5 structures, anatomical structures that are involved. Okay, or we can ask you that state all the systems that are involved or the structures that are involved for a typical reflex action to occur. Is that okay? Then you just state them: the stimulus, the sensory neuron, the interneuron, the motor neuron, and then the effector. Is that okay? So this is what we call the reflex action in nervous system. Is that okay? Great. And you see, this system is very important because it helps to protect your body from danger. Okay. Again, it also helps you to maintain posture and balance. Okay. And it also helps to regulate some body functions like blinking your eye, coughing. Okay. Some body activities like blinking of your eye and coughing. Is that okay? Yes. So this is what we call the reflex act. Okay. And pay attention to the timing over here. So a typical reflex action happens in about 0.1 to 0.3 seconds. Yes, so that is also a very important point that you have to note. Is that okay? Yes. So I have another image over here. It's the same thing. So here we have a hot object. So the hand is touching a hot object. So the pain receptors in the skin will pick the stimulus, and then it will be transmitted along the sensory neuron. So you see, from the sensory neuron, it moves to the relay neuron or the interneuron. Then the interneuron will do the processing. Is that okay? So from the interneuron releases it to the motor neuron. Then the information comes back to the muscle, which is the effector. Okay. Then the person will now move the hand away from the hot object. So you see over here, the person have now moved the hand away from the hot object. Okay. And this is how a reflex action occurs. So I've cited an example here, and I can ask you in exams to, with the aid of a diagram, describe the reflex act. You can write it down. With the aid of a diagram, describe a typical reflex action with the aid of a diagram. Then you use typical examples like this. So you can cite an example that, okay, when someone touches a hot object, is that okay? Now the sensory receptors in the skin will pick the information, and then the information with the signal will be, or the impulse will be transmitted along the sensory neuron to the relay neuron or interneuron, where the processing of the information takes place. Okay, then immediately response is sent back to the effector or the part of the body via the motor neuron for an action to be taken. Is that okay? At the effector. Okay, and this is how you do the description of a reflex act. Okay. That is fine. That's fine. So now let's look at the central nervous system. Let's look at the central nervous system. Okay, good. So now we are now going to remember we mentioned earlier on that the central nervous system consists of what? The brain and then the spinal cord. And then we look at the two divisions of the nervous system. We look at the central nervous system and then the peripheral nervous system. Now we are going to take each division one after the other. Okay, so we are going to start with the central nervous system, the CNS. Okay, so we are going to start with the CNS, which is the central nervous system. So remember that the central nervous system, the main structures over there is the brain and the spinal cord. Now, let me point out directly the anatomical features that we want. Okay, so the central nervous system consists of the brain. Now, the brain is about 1.4 to 1.5 kilogram, and the spinal cord is approximately 35 to 40 grams. Okay, and they all together, they make up less than 2% or about 2% of your total body weight. But they account for about 98% of your nervous system total mass. Do you know what that means? That means that if the entire nervous system is 100%, the brain and spinal cord alone is 98%. It tells you that the peripheral nervous system is something small. Is that okay? That is what it means. Okay, but if you are looking at the entire body, it's about 2% of your total body weight. Is that okay? And we are saying that the brain and spinal cord accounts for about 98% of the nervous system total mass. That is also something examinable that you have to pay attention to. Okay, now while the peripheral nervous system contains most of the nerve fibers, we have a lot of nerve fibers in the peripheral nervous system. But the main processing center for all incoming and outgoing informations, okay, are within the central nervous system. Now, if I ask you the main structures of the central nervous system, that is the brain and then the spinal cord. So the main structures of the central nervous system, or the main parts of the central nervous system, is the brain and then the spinal cord. Okay, great. So when we talk of the brain is responsible for memory, it's responsible for emotions, it's responsible for sensory, it's responsible for interpretation, it's responsible for coordination, okay, and vital functions such as breathing and heart rate. Now, the spinal cord is also responsible for transmitting informations between the brain and then the peripheral nervous system. Okay, it's responsible for transmitting informations between the brain and then the peripheral nervous system. Now, we have two types of tissues in the central nervous system. We have two tissue types in the central nervous system. We have gray matter and then we have the white matter. Now, you see, when we say a white matter, when the axons, anytime the axons, so in the remember, in the central nervous system, we have neurons. It's neurons that forms the brain. Remember, let me take that again. It's neurons that forms the brain. Okay. Now, when the axons of the neurons forming the brain, when they come together, they form the white matter. Okay. And again, the reason why we call it white matter is because, you see, most of the neurons in the brain, they are myelinated neurons. And myelin is made up of fat. And fat in the body is whitish in color. That's the reason why we call it white matter in anatomy. Okay, let me take that process again. Now, you know what forms the white matter is the axons of the neurons. So when the axons, remember the brain is made up of neurons. So when the axons in the neurons, or the axons in the central nervous system, when they come together, they form the white matter. But remember that most of the axons in the CNS, they are myelinated axons. And myelin is made up of fat. And fat in the body is whitish in color. That's the reason why the white matter, we call it white matter. Okay, because they are made up of axons. And axons in CNS, most of them are myelinated. And myelin is fat. Fat in the body is also whitish in color. That's why we call it what? The white matter. Okay. Now, the gray matter is also formed when the cell bodies, okay, so when the neuronal or neuron cell bodies of the neurons in the CNS come together, they form the gray matter. Is that okay? And because they are unmyelinated, because you see, the cell bodies are not myelinated, and that is the reason why they are gray in color. Okay, this is what we are talking about. Let's go to this diagram. This will help you. So you see, the gray matter is here, and then the white matter is somewhere here. Okay, so the white matter you are seeing over here, the reason why it's white is because the axons, most of the axons, the neurons in the CNS, that's the brain and spinal cord, their axons are made up of myelin. Their axons have myelin sheets. And you know, myelin is made up of fat, and fat in the human body is whitish in color. That's why here we call it the white matter. So you see, we've labeled here white matter. It should tell you that it's the axons, the myelinated axons of the neurons that are forming the white matter. And we can ask you that in anatomy. Why do we call it a white matter? It's a white matter because it's the axons of the neurons in the CNS that is forming it. And most of the axons in CNS are having the myelin sheets. And myelins are made up of fat. And fat in the human body is whitish in color. That's why we call it what? The white matter. Now, when you come to the gray matter, you know, what forms the gray matter? What did I say? What forms the gray matter? What forms the gray matter?
The neuron cell body.
Come again.
The neuron
cell bodies.
Okay, so the neuron cell bodies. Now, the cell bodies, are they having myelin sheet around them?
No.
No, they are not having myelin sheet. That is why they are not whitish in color. They are rather gray. Okay, so the gray matter is made up of the neuron cell bodies, and they are not myelinated. Neuron cell bodies are not myelinated. Okay, so in central nervous system, when you hear gray matter, then you remember that, oh, okay, gray matter is the neuron cell bodies in the CNS that forms the gray matter. And the neuron cell bodies, we don't have myelins around them. The myelins are only around the axons. Then that will help you to remember why they are gray in terms of their name. So that's why we call it gray matter. Okay, so they are not whitish because the cell bodies are not having myelin sheets around them. Then when you hear white matter, you ask, okay, why do we call it white matter? White matter because the white matter is formed by the axons of the neurons in the CNS. And the axons, most of the axons in CNS, most of the neurons in CNS, the axons are having myelin sheets. And myelin is made up of fat. Fat is also whitish in color. Is that okay? Great. So now my question. What is the difference between gray matter and white matter anatomically? And why do we call it white matter? Why do we call it gray matter? Who can answer that?
We say the white matter is made up of the myelin sheet in the CNS.
Excellent.
And the myelin sheet in the CNS is white.
Good.
Color,
Good.
and that is
because they are made up of fat. Yes. Excellent.
Fat.
Good.
And the
How about the gray matter?
It's made up of cell bodies and is unmyelinated.
Good. The cell bodies are not myelinated. And of course, a cell body, a soma is not having myelin around it. No neuron have the soma myelinated. Okay, and that's why we call that the gray matter. Okay, and this is a term that you'll be hearing along the line. And these are the two main tissues in the central nervous system, the gray matter and then the white matter. When we say gray matter simply means that when the neuron cell bodies in the central nervous system come together, they form the gray matter. And they are gray because they are not myelinated. They are unmyelinated fibers. And that's the site for processing of information. The axon is where the processing of information and decision making in the neuron take place. Okay. Then the white matter. Why do we call it white matter? White matter because what forms the white matter is the axon of the neurons. And most neurons in CNS are having myelin sheets around the axons. Okay, and myelin sheet is made up of fat. Fat is also whitish in color. That is why we call it the white matter. Is that okay? Please, are we together?
Yes, sir.
Yes, sir.
Okay, that is fine. That is fine. So you see, this is how the brain looks like. This is the central nervous system. So you see, we have the brain. Okay, the brain typically, so we have the brain, we have the cerebrum, we have the cerebellum, So this is the cerebrum, and then we have the cerebellum. Okay, so like I said, the central nervous system is made up of the brain and the spinal cord. So this is the brain. This one here is the spinal cord. Okay, good. So here we have the brain over here. We have the cerebrum. We have the cerebellum. We will look at more additional parts of the brain. And then we have the spinal cord over here. Okay, this is what we call the central nervous system. Is that okay? Now, let me quickly talk about this. Now, around the brain, okay, just around the brain, we have a membrane. The first layer of membrane around the brain and spinal cord. We call it pia mater. Say pia mater. Say pia mater.
Pia mater.
And that is the first layer around the brain and spinal cord. Okay, the pia mater. That is the first membrane around the brain and spinal cord. Okay, now we have 3 membranes surrounding the brain and spinal cord. Let me take it again. We have 3 membranes surrounding the brain and spinal cord. The first membrane, we call it the pia mater. And then the second layer, we call it the arachnoid matter. Say arachnoid matter. Say arachnoid matter.
Arachnoid matter.
Last membrane, we call it dura mater. Say dura mater.
Dura matter.
In Greek, when we say pia simply means something that is soft. Okay, so when I tell you that you are pia, it means you are soft. Okay, now in Greek, when we say dura means something that is strong. So you can use it as a term in your day. You can say I am dura. It means you are strong. Okay, now this dura is the membrane that is attached to the skull bone. This dura is the one that will be attached to the skull bone. Okay, or within the spinal cord, this dura is the one that will line the vertebral column. Okay, but this one is the one that immediately covers the brain. That's the pia mater. We call it, pia means soft. Is that okay? Pia is what we call soft. Okay, now in between this pia mater and arachnoid mater, that is where we have the cerebrospinal fluid. Okay, so remember that I said we have how many layers of membrane do we have covering the brain?
3.
How many layers of membrane? Okay.
We have 3 layers.
From the inside, which one is the first layer? From the inside, which one is the first layer?
The dura mater.
Hey.
The pia mater.
From the inside, which one is the first layer? Is the pia mater. Okay, which one is the middle layer?
Arachnoid matter.
Arachnoid mater. And which one is the last layer?
Dura mater.
Which of the layers is connected to the skull? The inside of the skull.
The pia
mater.
Hey, madam. The pia mater is covering the brain directly. And get it too. The pia mater is the one that covers the brain directly. That's the first layer from the inside. Okay. The one that is lined inside the skull is what we call the dura mater. Is that okay? So from the skull, which one is the first layer? Which one will come first from the skull?
The dura mater.
The dura mater. Okay, from the skull, which one will come last?
The pia mater.
The pia mater. Which one is directly in contact with the brain? Which of the layers is directly in contact with the brain? The pia mater. The pia mater. Which one is the middle layer?
The
arachnoid.
One is directly in contact with the skull.
The dura mater.
The pia mater.
One is directly in contact with the skull.
Dura mater.
Okay, which one is directly in contact with the brain and the spinal cord?
The
pia mater. When we say pia, what does it mean?
Soft.
When we say dura, what does it mean?
Something strong.
Something strong. So in Greek, when we say dura, it means something strong. Okay, where do we have the cerebrospinal fluid?
In
Where do we
between.
have the cerebrospinal fluid?
In
Arachnoid
between
matter.
the pia matter and arachnoid matter.
Come again.
In
Come
between the pia matter and arachnoid matter.
That is excellent. In between the pia matter and arachnoid matter. Now, listen carefully. This very one you have to know. This very one you have to know where we have the cerebrospinal fluid. That one you have to know. Because for the purpose of what you do in your next level, like medicine, for example, in advanced nursing too, you learn about lumbar puncture and those things. You have to know where we can find the cerebrospinal fluid in between the pia matter and the arachnoid matter. Is that okay? All right, that is fine. So the layers of membrane, I can ask you to write, okay, what are the layers of membrane? And the three layers that we've discussed in anatomy, that is what we call the meninges. Say meninges. Say meninges. So when we say meninges, meninges simply means the membrane surrounding the brain and the spinal cord. Okay. So when we say meninges in anatomy, it simply means the three membranes surrounding the brain and the spinal cord. What are these three membranes? The pia mater, the arachnoid mater, the dura mater. Okay, so if I ask you that, okay, state the three membranes or list the three membranes surrounding the brain and spinal cord, then you just write the pia mater, the arachnoid mater, and then the dura mater. Is that okay? All right, that is fine. Now, let's begin with the spinal cord. So this is how the spinal cord looks like. The spinal cord, this is how it looks like. Is that okay? This is how the spinal cord looks like. Okay, yes. So this is the spinal cord. This is how it looks like. Okay, great. So now let's go back to this point. Let's go back again. So we have the spinal cord. This is how it looks like. Okay, you can also look at it from this diagram. This is what we call the spinal cord. Okay, if I ask you to write the shape of the spinal cord, what will you write? How does it look like in your own words? What's the shape of the spinal cord? Yes, what is the shape from what you are seeing? This is the spinal cord from here. This is what we call the spinal. How does it look like?
Like a nail.
It looks like what? You said it looks like
A nail.
it looks like a nail. Hey, are you sure? Then I'm seeing something different too. I'm not seeing it like that. Okay. Anyway, it's also good.
Chain.
It looks like what?
A chain.
It looks like a chain. Okay, I think that is better. Yes, I think that is better. Okay, so the spinal cord, basically the spinal cord is cylindrical nervous tissue. It's like a cylindrical nervous tissue. That is what we say in anatomy. Okay, so it's like a cylindrical nervous tissue, and it extends from the foramen magnum, that's the base of the skull, to the L1 vertebrae in adults. Okay, so let me show you what we are talking about anatomically. So this space, if you remember in skeletal system, you were taught what we call the foramen magnum. Say foramen magnum. Say it after me. Say foramen magnum.
Foramen
Magnum.
Foramen magnum.
Over here is it. Here is it. That's the same thing we are seeing over here. So, and this is where, so you see, the spinal cord is connected to this process. It's connected inside. That is where the spinal cord, and this is the base of the skull, the base. Is that okay? So the spinal cord extends from the foramen magnum, that's the base of the skull, to around the L1 vertebrae in adults. So let's go back to the diagram. So you see, this is it. This is what we are talking about. So you see, the L1 vertebrae is somewhere here. So the spinal cord starts from here. The foramen magnum is somewhere here in the base of the skull. So it extends from here down to about the L1 vertebrae. Is that okay? Especially in adults. But that doesn't mean that is the level it ends to. It continues. It goes down. Is that okay? Yes. So,
Sir,
but
please,
practically,
no. Sir, please, can you do
Sir,
the
please thank you.
pointing again?
oh, this is what we are talking about. So we are saying that this is the foramen magnum. This is the base of the skull. And we are saying the spinal cord is connected here. And if you are looking at it, it's like this. So it's connected. So the spinal cord is connected to the base of the skull. And through the base, it connects to the brain. Is that okay? Yes. So it's
Yes,
like this.
please.
So it's like it's connected. That is where the process is. So it's connected here. The base of the skull here. Is that okay? And that is what is shown over here. The foramen magnum. So the foramen magnum is somewhere here. Is that okay? So it starts from this point to about the L1 vertebrae. The L1 vertebrae is somewhere here. Okay, but what I'm saying is that in most anatomy books, they will tell you it starts from the foramen magnum to the L1 vertebrae. But that does not mean that is where the spinal cord ends. That does not mean that is where the spinal cord ends. Okay, it continues and go down. Okay, good. So we will look at all the subdivisions of the spinal cord. Okay, great. So we say in anatomy, we say that it is divided into 3 regions. We have the cervical region, we have the thoracic region, we have the lumbar region, and we have the sacral region or segment. So we have the cervical segment, the thoracic segment, the lumbar segment, and then the sacral segment. Okay, great. Now, if I ask you that, where does the spinal cord begins from? What will be your answer? Anatomically, where does the spinal cord begin from?
Of the skull.
It begins or it starts from the foramen magnum. That one is very important. You have to know. It's very important. Okay, so that one is very important. You have to. Now, again, another thing I can ask you is about the segments of the spinal cord. Okay, practically, we have about 4 segments. So we have the cervical segment, we have the thoracic segment, we have the lumbar segment, and then we have the sacral segment. And this is what we are talking about. So you see, we have the cervical region or segment, the thoracic region or segment, we have the lumbar region or segment, we have the sacral region. In some books, they will even go further and even talk about the coccygeal segment. Okay, so here, so you see, it starts from here, the cervical region. The cervical region starts from the C1 to C3 to C7 to C8. Okay. Now the thoracic region starts from T1 to T11 L2. Is that okay? Then we have the lumbar region. The lumbar region starts from L2 to S1. Then the sacral region starts from S2 to S3. Then we have the coccygeal segment. Is that okay? Yes. So these are the regions of the spinal cord. Okay. So we have the cervical region, the thoracic region, the lumbar region, the sacral region, and then the coccygeal region. Now, each of these regions and the part of the body they control. Please listen carefully. Each of these regions and the part of the body they control. Okay. And you know the spinal cord, the nerves from all parts of your body are connected to the spinal cord. So each of the regions on the spinal cord has a part of the body they control. For example, at the cervical region, especially the C1 to C3, they control your neck muscles. C4 controls your diaphragm. C5 controls your deltoid or shoulder. C6 controls your wrist. C7 controls your triceps. C7 to C8 controls your fingers. Okay, then we come to the thoracic region. The thoracic region, T1 controls your hand. T2 to T12 controls your intercostals and the trunks. T7, L1 controls your abdominals or abdominal structures or organs. T11 to L2 controls is responsible for ejaculation. Okay, then we come to the L, the lumbar. The lumbar, L2 controls your hips. L3 controls your quadriceps. L4, L5 controls your knee. L4, X1 also controls your foot. Then we have the sacral region. The S2 controls your penile erection. Okay. Then the S2, S3 is responsible for bowel and bladder functions. Is that okay? Yes. So all these things you have to know. They are key anatomical things. Don't play with them. Don't play with them at all. And I have a way that I'll make sure you will learn these things. You have to know these things off head. Is that okay? Yes. So the cervical region, you should know. So every day, look at it. Every day, look at it. So the cervical region. But ideally, you can learn it you can summarize it and say that, oh, the cervical region controls the neck muscles, the diaphragm, the deltoid, the wrist, the triceps, and the fingers in a summary. Then you can say the thoracic region controls the hand, the intercostals, the abdominals, and is responsible for ejaculation. Now, the lumbar region controls the hips, the quadriceps, the knee, and the foot. The sacral region is responsible for hot pineal erection, okay, bladder and bowel functions. Okay, and I mentioned that some books will even go further and talk about the coccygeal region. And the coccygeal region is the region around your tailbone. Okay, to some extent, they are also partly responsible for your bowel and bladder activities in a way. Okay, so that's the coccygeal segment. Okay, good. And these are the regions of the spinal cord. Please, this area, like I keep repeating, is very, very important to us in anatomy. Okay, so everything about this slide that you are seeing, please take it very serious. Is that okay? Take it very serious. Okay, good. So the cervical region, we have the C1 to C8. They innervate your neck, the shoulders, the arms, and the diaphragm. So they are here. The thoracic region, about 20 centimeters. You should also know the centimeters. So the length of the cervical region, the length of the thoracic region, the length of the lumbar region, the length of the sacral region, the length of the coccygeal region, which is less than 1 centimeters. Okay, so the cervical region is 13 centimeters, the thoracic region is 20 centimeters, the lumbar region is 6 centimeters, the sacral region is 5 centimeters. Is that okay? Yes, so pay attention. Make sure you get everything here. The regions of the spinal cord. Okay, great. So maybe what you can do is that you can draw it. Okay, you can draw it yourself. Then every day you look at it, you draw it. Every day you look. These ones we are not interested in, but fine. At least know the functions of the spinal cord for conduction of impulse. Of course, for locomotion, it also plays a role in your ability to move and walk. And then also, you know, the spinal cord is what functions in the reflex action. You know, in reflex action, spinal cord plays a role. Okay, so reflex action, we have the stimulus, the sensory neuron, the relay neuron is inside the spinal cord. Then we have the motor neuron, then the effector. So spinal cord plays a role also in reflex action. Okay, that is also something you can talk about. But we are not so interested. Now, let me point on the key anatomical things we are interested in. Now, one, know where the spinal cord extends from and know the shape of the spinal cord. That is one. Know where the spinal cord extends from the base of the skull, specifically the foramen magnum, and know the shape of the spinal cord. Okay, again, know all the divisions of the spinal cord. So the cervical region or division, the thoracic division, the lumbar division or lumbar region, the sacral region, and then the coccygeal region or coccygeal segment. Is that okay? So when we take the cervical region, what is the centimeters? 13 centimeters. You should know the part of the body that is innervate. And tomorrow when I come to class in the morning, we will do this exercise. Okay, when I call you, you tell me the region, you tell me the organs that it controls. Okay, and I can also mention, okay, C4, which part of the body does the C4 level of the spinal cord control? Then you tell me diaphragm. Okay, T2 to T12, which part? Intercostals. Okay, L3, which part of the body does it control? The quadriceps. Memorize it. Make sure it's part of you. Is that okay? And when I meet you outside, I can ask you. Okay, yes. If I meet you outside and I notice you, I can ask you that the L2 level of the spinal cord, which part of the body does it innervate? Then you tell me the hips. Is that okay? Yes. So that is fine. So all these things, you have to memorize all of them. Is that okay? Great. And in exams, we can bring about 5 of them. We can randomly select some of them. C6, C7, L2, L4 to L5. We will miss them then you will write out the parts of the body they innervate. Okay, so you have to memorize and make sure you are sharp when it comes to these things. Okay, great. So that is it for the spinal cord. These are feather diagrams. It's the same thing, but this is another diagram. Okay, so we have the cervical region, the thoracic region, the lumbar region, the sacral region, and the coccygeal region. These are feather. It's just a new diagram, but it's the same thing. All right, that is fine. That is fine. So now let's get to how the spinal cord looks like. Okay, so the cross-sectional view, this is how the cross-sectional view of the spinal cord looks like. I think it's a good ground for us to end over here today, so that we can continue. Sorry? Come again? Hello? Hello?
It's fine.
Can you see the slide now?
Yes.
Okay, that is fine. So the cross-sectional view of the spinal cord, this is how it looks like. And you see this diagram you are seeing over here in the slide. This one, it can come in exams. We can draw it and ask you to label. The diagram you are seeing over here. We can give you the diagram just like you are seeing. Then we'll ask you to label it. It's an examinable area that we ask questions on a lot. And this is the cross-sectional view of the spinal cord. This is how the spinal cord looks like. Is that okay? This is how the spinal cord looks like. And I'll walk you through the various parts. Okay, it's very simple. I'll walk you through the various parts. But practically, I'll just introduce you to it. Then when we meet in person tomorrow, we will continue. It's very possible in our first 1 hour tomorrow, we can finish with the nervous system and start with the endocrine system. Is that okay? All right, that is fine. So, yes, so let me just, do we have Lawrence over here?
Yeah, he's off.
Okay, that is fine. So you see, we have here is the gray matter. So I've started introducing you to the cross-sectional. This is the cross-sectional view of the spinal cord. So as we are learning, you are paying attention. Okay, so over here, you see, so over here, if you check, and as we move on, I'll be showing you how you can use it to understand the. So this is what we call the white matter, and this is the gray matter. Okay, this is the white matter, and this is the gray matter. And I've told you what the white matter is and what forms the gray matter. Now, if you come over here, this one is the gray matter. So look at it. This one is the gray matter. Can you see what is looking like the butterfly portion? This is the gray matter. And this one is the white matter. Okay, now the gray matter has parts. Okay, so you see this part of the gray matter here, we call it the dorsal horn. Say dorsal horn. Say it after me, dorsal horn. Yes, dorsal horn. So you see over here to his here. You see over here to his here. Dorsal horn. Okay, say dorsal horn. Say it again. Say dorsal horn. What is another term that we give for dorsal? What is another term for dorsal? Dorsal means posterior. Have you forgotten? Dorsal means posterior. Is that okay?
The back.
Yes, the back or posterior. Is that okay? Okay. Then here we call here the ventral horn. Say ventral horn. Say it after me. Ventral horn.
Ventral horn.
For ventral horn.
No, like in front.
For in front.
Yeah.
You can also call it the anterior horn. Is that okay? Anterior horn. And this one is called the lateral horn. So see, the gray matter is divided into 3. We have the dorsal horn, the lateral horn, and the ventral horn. Okay. So can you see that the gray matter is divided into 3? We have the dorsal horn, the lateral horn, and the ventral. Or you can say the gray matter, or you can say the gray matter has 3 parts: the dorsal horn, the lateral horn, and the ventral horn. Now I'm coming to point another part of the gray matter. Then you tell me the part. Which part is this?
So we can't see your arrow.
Horn. Okay, which part is this?
Dorsal.
Which part is this?
The
The
lateral.
Lateral
lateral
horn.
horn. Lateral.
Oh, no.
Which part is this? Which part is this?
The lateral.
Part is this? The lateral horn. And which part is this?
The ventral.
Ventral.
Ventral horn or
Anterior
The
horn.
anterior horn.
anterior horn. Can you see that there is something like an innervation over here? It's like a demarcation, more like a line, an inner demarcation over here. Can you see? It's like a line. Yes, we call it the
Yes.
dorsal median sulcus. Say dorsal median sulcus.
Hello.
You can see. I think you should go off and rejoin because you are the only one who cannot see. Yeah. So, and it happens that we just go off and rejoin. Try and rejoin and see if it works for you. Okay. All right. That is fine. So here we have the dorsal median sulcus. Say dorsal median sulcus.
Dorsal median sulcus.
Median sulcus. Say it again. Dorsal median sulcus.
Dorsal
Median
Median sulcus.
The last time. Say dorsal median sulcus.
Dorsal median sulcus.
In front of the cross section too, we have another inner demarcation here. We call this one ventral median fissure. Say ventral median fissure. Say it after me. Ventral median fissure.
Ventral median fissure.
Then let's go again. Say dorsal median sulcus.
Dorsal.
median sulcus.
Say it. Ventral median
Ventral median sulcus.
See whether let's go to the diagram and let me see whether you are learning. Okay. What is the name given to eye? Look at eye. What name will you give to eye?
Dorsal median sulcus.
This shows that you've learned. Okay, what name will you give to the P? P. Look at where P is pointing. Look here. What name will you give to P?
Ventral median
Fissure.
Fissure. Okay. Say it again. Ventral median fissure. Say it again.
Ventral median
This shows that you are learning. Yes, this shows that you are learning. Is that okay? This shows that you are learning. Now here, what is the name given to this part? Here, where I'm pointing now. Here. The eye.
The dorsal median sulcus.
To K. Look at K. What name is given to K?
Dorsal horn.
Posterior horn.
Horn. Okay. What name is given to M?
The lateral horn.
What name is given to L? Yeah, horn. Okay. When we put K, L, and M together, what is the name?
Gray.
You are smart. That is what we call the gray. Portion. We call this portion hear me? Hello?
Why is not good, please?
Can you hear me?
Please,
No,
yes, we
sir.
can
The
hear you
line
now.
is breaking.
Say posterior funiculus. The A. Say posterior funiculus.
Posterior funiculus.
The B is anterior funiculus. Say anterior funiculus. Say anterior funiculus.
Nicholas.
Then the C, we call it the lateral funiculus. Say lateral funiculus.
Lateral
Lateral
funiculus.
funiculus.
All the funiculuses come together to form the white matter. Okay, so let's go again. So say it after me. Say posterior funiculus. This one. A is posterior funiculus. Say posterior funiculus.
Posterior funiculus.
Funiculus.
Funiculus. Anterior funiculus.
Anterior
Funiculus.
anterior
funiculus.
Lateral funiculus.
lateral
Funiculus.
So what is D? What is D? D is a combination of all the three funiculus. So what is D?
white matter.
Ice matter.
Funiculosis forms what? All the funiculosis forms the white matter. Have you gotten it?
Yes.
And then the horns forms what? The horns will form what?
Gray matter.
The gray matter. This shows that you people are really concentrating.
We are
And then
learning.
you see there is a central, something like a canal over here. Can you see there's something like a depression, like a canal over here?
Yeah.
Like a who? We call it the central canal. Says central canal.
Central canal.
Central.
Or you can call it the spinal,
Spinal.
or you can call it the spinal canal. Say spinal canal.
Spinal canal.
Central canal or spinal canal. Now we are almost completing. We will end with this diagram. We are almost completing. So now let's look at here. So you see the can you see the nerve over here? The nerve connecting to the spinal cord. Can you see a nerve is connecting? Now the nerve connecting, you see the nerve is divided into two. The nerve, if you look at it very well, you see it's divided into two. Now the part that is connecting in anatomy, we call it root. But you can see clearly it is two. Now the one at the back, we call it the posterior root or the dorsal root. Okay. You can call it the posterior root or you can call it the dorsal root. Is that okay?
Yes, sir.
Good. So you can call it the posterior root or the dorsal root. If this is posterior root, what name will you give to this one? The H. The anterior root or the ventral root. Is that okay? Good. The ventral root. Then you can see over here there is something like a bulge part of the nerve over here, the E. We call it the posterior root ganglion. Say posterior root ganglion.
Posterior root ganglion. Posterior root ganglion.
Posterior root ganglion.
Posterior root ganglion.
This part is called the spinal nerve. Now let me bring it over here. What name will you give to this one? The one I'm pointing now. What name will you give to this one? One. Is that the same thing we are doing here? What name will you give to this one? It's not true. This is posterior root. The spinal nerve is this one. The spinal nerve is here. This is the spinal nerve. This is the posterior root. What name is given to the part I'm pointing now? Here. The root I'm pointing. What name is this?
Anterior root.
The anterior root. Okay. What name is given to the swelled part that I'm pointing? This portion.
Ganglion.
Posterior root
Ganglion.
Posterior root
Ganglion.
So now we are going to complete the entire table. Then I can confirm that you've learned. So I'll point the part, then you tell me the name. Okay. Which part is J? Which part is J? Which part is J? J.
Posterior horn.
Which part is J?
Dorsal median sulcus.
Not true. The J is Gray Cominiger. Say Gray Cominiger.
Gray comb.
It's for you to see. The J is this part.
Okay.
So you see this part. This part is what we call J. So it's like the curved part of the curved part here. Not the canal. It's on top of the canal. Just a little on top of the canal. We call it the Gray Cominiger. Say Gray Cominiger. Say it again.
Great
Gray
communism.
Cominiger. For the last time, say Gray Cominiger.
Great communism.
So let's go back to the diagram. Okay, which part is I? Which part is I?
Dorsal or posterior?
Dorsal median sulcus.
Posterior median sulcus.
So the dorsal or posterior median sulcus. Okay, which part is P? P. P. P.
Ventral
Sulcus.
fissure.
The ventral or anterior median fissure. Which part is O?
Ventral spinal
Ventral
canal.
canal. Or
Spinal.
Spinal
That is
canal.
excellent. That is excellent. Okay, which part is C? Which part is C?
Lateral funny close.
Close.
posterior
Panisculus.
Panicles. Which part is A?
Posterior.
Panicculus.
When we put A, B, and C together, what do we get?
Right
matter.
Okay, which part is E? E. 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 E.
Root ganglion. Ganglion.
F. F.
No.
On the labels.
Oh.
Okay. Which part is J?
Posterior root.
The posterior root. Okay. Which part is H? H.
Anterior root. Ventral root.
Ventral root.
Okay. When we put J, K, L, and M together, what do we get?
Gray matter.
Gray matter. Okay. Which part is K? Which part is K?
Posterior horn.
L.
Horn.
Which part is M?
Lateral horn.
Which part is S?
Dura matter.
Dura matter. Which part is Kill? Kill. Pia matter. And which part is the R?
Arachnoid matter.
Arachnoid matter. Please remember, this is a potential diagram for your exam.
Okay. ABC there up to S.
A potential diagram for your exams. And once you finish labeling something like this, I'll assign 20 marks to it. Once you do the complete labeling, 20 marks. Is that okay? So you can note today's dates down. This is a potential diagram for your exams. Yes, sir. It's a potential diagram for your exams. Okay, so you can start practicing it. I wouldn't let you draw this diagram in exams. I will just bring the diagram, then you label the parts. Is that? I'll bring the diagram, then you label the parts. Another thing I can do is that I can label all the parts wrongly. Then I'll ask you to identify the wrong labels. Is that okay? All right, that is fine. Yes. So I think we can end over here. We can end over here.
Sir, please don't label the parts wrongly.
Oh, you don't want the parts to be labeled wrongly.
Yes.
Yeah.
You don't want that one.
On that one.
It to be drawn, then you label the part yourselves.
Yeah.
Okay, that is fine. That is fine. All right. So I wish you all the best. We will meet tomorrow, God
Thank
willing, and
you.
then we will finish with the rest of the slides. Oh, the slide is with your rep. The slide is with your rep already. You can see him and get a copy for yourself.
Thank you very
That is
much.
fine.
You
That's
say.